It's my pleasure to introduce Dr. Vlad Ayzenberg as today's Vision Seminar speaker. Thanks, Vlad, for accommodating a late invite request to get the fall seminar going. So Vlad received his PhD in psychology and neuroscience at Emory University, where he worked with Stella Lorenko, working on the origins of object and spatial representations. He then did a postdoc at Carnegie Mellon, working with Marlene Behrman, studying development and neural bases of visual object recognition, and then joined Penn as a both Mindcore and Data-Driven Discovery Initiative Fellow. During his time at Penn, I had the privilege of working with Vlad, collaborating on projects looking at the structure and function organization of the human newborn visual system. Earlier this year, Vlad joined Temple University as an assistant professor in the Department of Psychology. His research bridges psychology, neuroscience, and machine learning, focusing on how humans perceive, learn, and represent the world from infancy through adulthood. His lab focuses on three main themes: how infants demonstrate early abilities, such as number sense and intuitive physics, how people rapidly acquire new concepts from only a few examples, and how the brain forms stable object representations that support recognition across changing contexts. This impressive body of work Vlad has made very influential contributions and has had many impactful papers in journals like Science Advances, Current Biology, Nature Reviews Psychology. Today, he's going to share his work on the building blocks of visual cognition, focusing on how early developing systems of the brain support flexible learning and perception. So please join me in welcoming Vlad.
So since it's mostly our labs, I think we can make this pretty informal and we can talk about some data that we have literally coming in that Alan is just working on analyzing and how that relates, and maybe think about where some of this work can go, or I'll talk a little bit more about where some of this work is going, and maybe some other ideas about kind of how to build out this research program. This was originally my job talk that I've sort of adapted for every talk now. And I've sort of been adding pieces to it as I go. And I'm starting to do the thing where the faculty thing where you work on your slides right before you give a talk. So we'll see. Hopefully they come up smoothly. Okay, so title of the talk is Early Development Mechanisms Underlying Visual Object Categorization. And so, we know, it's a little delayed. So we know that the human visual system has a really remarkable capacity for abstraction. So even though this cave painting is like 10,000 plus years old and bears few properties of the real world, we can still identify these cave paintings as containing people and animals, right? And this is something that even a young child could tell you what's in this image, even though they probably maybe haven't seen a cave painting before. And beyond that, we know that our visual system is extremely powerful and is able to interpret visual information quickly and effortlessly. For example, here I'm showing images presented for just 70 milliseconds, so less than a tenth of a second. And it's still pretty easy for you to identify these objects as they appear. And as the cave paintings illustrated, the specific input needed to recognize these objects need not bear any resemblance to the real world. Right? You probably have never seen a wavy lamp or a wavy airplane. This image has never hit your eyeball before, but you can still recognize this. And this type of rapid object recognition isn't just easy for adults. It's also something that kids seem to be able to do really well. So we actually gave preschool children an even harder version of this task, where images could be presented in these kinds of perturbed or deleted, kind of deformed conditions, up to 100 milliseconds and with a forward and backward mask. Right? So this is, we thought would be pretty hard for young kids. But we found that kids overall did really well on this task. Even 3 and 4-year-olds could identify these objects at speeds as fast as 100 milliseconds. And the 5-year-olds were practically adult-like at the slower speeds of 250 and 300 milliseconds. Right? So this tells us that these kinds of robust object recognition abilities arise pretty early in development. We may not need that much experience to have this sort of powerful ability. And what I think is most interesting about this is just how much experience is needed to accomplish this type of recognition. For example, we can compare the abilities to of humans to computer vision models such as deep neural networks. So what I'm showing you here is a plot of deep neural network performance as a function of how many images they were trained on. So these are various computer vision models that were trained on object classification tasks or maybe vision tasks more generally. And so what you generally see is that performance goes up the more training they have, right? This is kind of a typical power law in the neural network literature. Um, but where would humans be on this plot? So it's a little hard to compare model experience to human experience, but to give you just a sense of the scale of these numbers of what 10 billion images really means on the human lifespan, we can sort of give you this not metaphor, this little snippet of what that means. So imagine if from the moment of birth, a child were to see a new image every 100 milliseconds of their life. So this is faster than a saccade, right? A typical saccade is 200 milliseconds. So they see a new image every 100 milliseconds of their life without ever sleeping, so nonstop image presentation. They would not see as many images as a fully trained clip model until they're 40 years old. Right? So this is a crazy amount of training that these models are getting. And it's clear that humans don't need this much training. If we were to plot the human data using this metric, they fall to the left, up and to the left of the line, meaning that they are more accurate with far less data, right? And exponentially less data than these models. So again, this tells us that these object recognitions are robust from early development, and they might not need that much experience to arise. But of course, humans are not only good at recognizing objects, familiar objects like airplanes and lamps, we can also rapidly learn new kinds of object categories. For example, I can show you an object maybe you haven't seen before, like this instrument called a sheng. We're actually now doing experiments with these novel objects, and so this is one of my favorites. And you would readily be able to identify other examples of a sheng and do so across these different kinds of contexts. So, how do we make these kinds of generalizations with so little experience? Um, this is the broad question that my work and my lab is trying to answer. And specifically, we're interested in understanding the computational and neural mechanisms that might support these kinds of generalization abilities, and critically, how they might arise over developmental time. So in this talk, I'll primarily focus on the problem of object categorization. That is, how do we rapidly learn a new object category? But as Mike kind of alluded to in the introduction, this issue is not just relevant for object categorization, it's also relevant to a host of cognitive skills that seem to arise really early in development, things like intuitive physics, numerical cognition, and so on. But for now, let's start with object categorization. So to give you a bit of a roadmap for the rest of the talk, in the first study, I'll use eye tracking modeling to ask what kinds of representations support novel object categorization in a population that has very little experience, namely infants. And then using fMRI and high-density EEG with adults, I'll examine what neural mechanisms might support these kinds of categorization abilities, at least in the mature brain. And finally, in the last set of studies, I'll use fMRI with newborn infants as well as patients to ask how do the neural pathways underlying categorization develop. And of course, please feel free to interrupt me at any point. Okay, so let's start with this first question. What visual representation might support object categorization in infancy? Well, to go back to the examples in the very beginning or a minute ago, we know that we can identify objects when they have few properties of the real world, like these cave paintings, and that we can recognize exemplars of a category even when they're new and share few local visual features in common. So this tells us that we don't rely on any particular visual feature to recognize the object, but might recognize these objects on the basis of some kind of more abstract object property, something that isn't encoded in the pixel values of the image. So what might that property be? Well, one possibility is that we form such object representations not by focusing on those visual features, but on their spatial relations. That is how those features are arranged. So, consider a common set of objects like this guitar. These guitars have very different features, but the overall arrangement of those features, where the neck and body of the guitar are relative to one another, is very similar. In fact, I can show you a new object, and you can still identify this object as having a guitar-like shape. But if I simply rearrange the pieces of that object, give it a new spatial configuration, it becomes a totally different object. This might feel reminiscent of how David Marr initially described human shape perception, not as requiring a detailed description of object surfaces or features, but as a structural description that described where those features were relative to one another. And so we can see how spatial information might be used to form this kind of abstract global shape representation that supports categorization. Um, but now we want to know, is this a cue that we use early in development when experience is more limited? Do infants categorize objects using the spatial structure of an object? And so to test this, we recruited 84 6 to 12-month-olds, and we tested whether infants would categorize objects on the basis of their spatial relations across a change to their local feature. So you can see that these two objects have the same underlying spatial structure, so kind of an arm sticking out from either side of a central body, but their local features, their surface features, differ between the two. And so we specifically tested this age group because we know that object experience is really limited at this age. Just 10 objects, 10 unique object types, occupies about a third of infants' visual experience. So these objects should be totally novel to infants. They shouldn't really have any expectations about how these objects might vary within a category. And so we tested whether infants would categorize these objects by their spatial structure, and we compared them to a series of other objects that had qualitatively different kinds of spatial arrangements of parts. Okay? And so to do this, we used what's known as a habituation-dishabituation paradigm. Infants were shown one object on the screen over and over until they essentially got sick of looking at it. Well, their looking time decreased to some criterion. And then we tested them with an object that had a familiar structure, but now a new set of local features. So went from skinny to these like kind of bulgy features, or an object that also had new features, but now had a different spatial structure. And so the prediction is that if infants generalize across a change to the object's local feature, then there shouldn't be any difference in looking between the habituation and the familiar object, but they would look longer with this object with this novel structure, even though both are different on the retina, right? They both are visually different. And importantly, we first made sure that infants could equally discriminate the objects, meaning we made sure that it wasn't just that they couldn't tell the difference between habituation and familiar, right? They can see that those are different, but now we're really asking about generalization, about categorization. And so we tested infants in two experiments, one where the structures were qualitatively different like this, and one where they were much more similar. Indeed, if you look closely, the main difference here is one of these arms kind of moved down the length of the body, right? This is now a much finer grained change in spatial structure. And it's also a nice control for image level properties because you can see the image level properties of these objects are much more similar on the retina. Right? So this is what we thought would be a pretty hard test for young infants. Um, and so what we found is that infants correctly classified objects by their spatial structures, suggesting that they were able to generalize across that local feature change. And this is a finding, oh, I'm sorry. And so they generalize across spatial structure, meaning again that they look longer at the object with the novel structure compared to the familiar structure, and there's no difference between that familiar spatial structure and the end of habituation, right? Suggesting they kind of treated those as similar. And we replicated this in the second experiment where the structures were even more similar, right? That they could do this. They were really sensitive to that change. So this tells us that infants were able to generalize across a change to the object's visual feature, but we could still ask, how was this accomplished? Were infants really categorizing objects by relying on their spatial structure, or maybe there's just some other visual cue that co-varies with these differences, right? Maybe they're doing something else. So to test this possibility, we compared infants' performance to a series of computational models. Specifically, we first compared infants against what's known as a skeletal model, and this is a computational model that describes the spatial structure of objects. So to kind of elaborate on this a little more, skeletal models or shaped skeletons are pretty intuitive. One can think about them as kind of like the stick figure of an object. So a stick figure provides a nice, accurate description of the human form without describing any of the surface properties of a person, right? Instead, it describes where those properties are relative to one another, where the arms and legs are relative to one another on the body, or for another object, where, say, the wings and body are relative to one another on an airplane. And so we thought this would be a good candidate model because A, it describes, it can accomplish object recognition using spatial relations. And we know from computer vision work, it's fairly invariant across exemplars within a category. So again, even though these guitars have different surface features, their underlying skeletons are still more similar to each other guitars than they are to any other category. And so in addition to a skeletal model, we also compared a series of control models. Specifically, we compared infants to a series of deep neural network models. Um, so I think this audience knows what deep neural networks are, and I've already mentioned them a little bit, but basically deep neural networks are computational models that might learn to categorize objects after many examples. And they do this by representing increasingly more complex properties of the object as you move up a hierarchy of layers. And so one of the reasons people are so excited about them is that they can do many object recognition tests humans could do using similar inputs and outputs. Meaning I could show them a picture of a dog and it would correctly classify it as a dog. And what makes them particularly interesting from a neuroscience perspective is that there seems to be a functional correspondence between the hierarchy of a deep neural network model with the hierarchy of the primate ventral pathway, the pathway most associated with object recognition. And so we compared infants to all of these models, and we specifically chose models that let us test different kinds of hypotheses. For example, we tested a model that had a recurrent architecture, much like the adult ventral pathway, a model whose training was intended to induce a shape bias by decorrelating local features, like local color and texture information, and a model that was actually trained on naturalistic experience from the head cams of videos, right? So this is a model that was actually given infant-like experience for its training. And so these models are now a few years old, but they're still generally very high-performing models, and they provide a good match to brain data. And so what we found when we compared infants to all these models is that infants' performance was best matched by the skeletal model, not any of the deep neural networks. And this is a finding that we replicated in the second experiment as well. And here the deep neural networks didn't even perform above chance. And so again, the skeletal model is the one that represents the spatial relations between parts. So these findings provide more direct evidence that infants may have categorized these novel objects, these objects they've never seen before, by relying on the spatial relations between object parts rather than some incidental visual feature. But we might ask from these data why the deep neural networks perform so poorly. These are supposedly some of our best models of human vision. What could be going on? Maybe these stimuli are just weird or our testing procedure is weird. And so to examine this possibility, we tested how well these models would categorize objects now using their local visual features here across a structural change. So in this case, the models were habituated to one object and then tested with objects that either had the same local visual features or different local visual features. And now both objects had different spatial structures from habituation to test, right? So this is kind of like flipping the test, right? Testing on feature instead of structure. And here what we found is that the skeletal model was bad at the task, couldn't do it, but now the deep neural networks were all above chance, suggesting that unlike infants, the deep neural networks were relying on the local visual features of the objects, not their structure, and essentially using qualitatively different kinds of visual information than the humans. And so to kind of briefly summarize this first section, what visual representations support object categorization in infancy? We found that categorizing novel objects may be supported by spatial representation of shape. Mhm. No, we did not. It's very time consuming. It just takes years to run that in infants. Um, but to speak to some data we're collecting now, we are running it in older kids and adults. Not that specific condition, but we're basically testing how human adults use shape and local features to categorize objects. So what you can do is you can kind of visually scramble the images to preserve the features but not the shape versus just show the shape. And we find that for familiar objects, adults can do both, right? And then for novel objects, maybe they can only use shape. So TBD. That's the data was collected last night. So we're doing this thing where we're training, we gave them these novel instruments that people haven't seen before, like that Shang, right? And then seeing, how quickly, basically teach them one example and then testing them on these different manipulations. And then we're also running that with like 4-year-olds to see. So So by adulthood, we can certainly use both cues, but maybe only for things that we're overly familiar with. Yeah, yeah. By 6 months, I don't think it interacts at all. By 6 months, their acuity is almost adult-like. Yeah, acuity gets better really quickly, and I would say even in newborn infants, right? Yes, their vision is poor, much poorer. It's like 2600 or so. But again, this is one of these things where it's hard to imagine what that looks like. At 2600, you can see a 12 centimeter stimulus 60 centimeters away, right? So you can still see this pretty clearly. So that's about the distance we test at, right? That's also the distance of mother's face, the distance of a lot of things, right? So their acuity, they're not blind. They can still see a lot. Yeah. Any other questions about the infant stuff? Cool. All right. So next we asked what neural mechanisms might support these kinds of categorization abilities, at least in adults. And so, decades of research has shown that object categorization is supported by this hierarchy of regions along the ventral visual pathway. But as I've already shown you, our best computational models of the ventral pathway, deep neural networks, seem to be missing this crucial aspect of recognition, namely a representation of the shape structure. So obviously we shouldn't assume the biology is wrong just because we might have some bad models. But in this case, there's actually a reason to think that the ventral pathway is much more like deep neural networks than we even previously believed. For example, there's accumulating evidence that the ventral pathway might also not represent shape information, but like neural networks, is better described as a basis set of local features. For example, a slight perturbation to an image can radically change the firing rate of ventral neurons. So if you have, this example neuron that seems to prefer, I guess, pipe gauges and not dogs, right? So the response to dogs is much lower than pipe gauges. But then add this really tiny image perturbation, which you could probably only barely see here in this second example. Um, you can radically change the firing rate of that neuron such that it now responds even more to dogs than it did to originally did to pipe gauges. So people familiar with neural net literature can think of this as like an adversarial example, right? This is what we do use with adversarial examples in neural nets. We do these slight statistical manipulations and we change the predicted class of a model. So this suggests that at the single neuron level, they're very sensitive to specific visual features in the image. But we also find this at the population code level, that the population code of ventral path might not be sensitive to shape either. So for example, if we were to present participants with images with intact shape or intact spatial relations versus these feature scrambled ones, so these are kind of the ones we're testing with now, we find that behaviorally, humans can certainly tell these apart, right? You know, you would group two examples of the scrambled images together and away from this intact one. But the representations of the ventral pathway as well as deep neural networks seem to treat all these equally. They're almost interchangeable. They can't tell the difference between the intact and scrambled versions. And so if the ventral pathway isn't sufficient to compute shape, what might we be missing? One possibility is that a global shape structure, a spatial structure, may be computed regions of the dorsal visual pathway, such as the interparietal sulcus or IPS. And then this information may be transmitted down to the ventral pathway to support categorization. So why do we think the dorsal pathway may play this role? Well, from classic research, we know that the dorsal pathway plays a key role in computing the spatial relations more generally, right? We know it's involved in spatial processing. And so one hypothesis is that the dorsal pathway may compute the spatial relations among features, much like the skeletal model. So it may compute, say, where those features are relative to one another in an object. So to test this, we conducted two experiments. We first tested whether the dorsal pathway represents the spatial relations of an object's features. And then we tested whether these regions are actually contribute to object categorization. So to test this first possibility, I created a functional localizer task to identify regions that were more sensitive to spatial relations, the spatial relation between features than the features themselves. So we had images where the features of the objects were all the same, so they might have had these like hourglass shapes on them, but the spatial relations between those features varied, sometimes on top, sometimes side to side. And that's contrasted with trials where the spatial relations stayed the same, so maybe they all have this T shape, but the features changed from trial to trial. Okay. And what we found is two regions in the dorsal pathway that were more sensitive to the spatial relations of features than the appearance of features. One in posterior IPS and one in anterior IPS. And these regions were only in the right hemisphere, or at the group level at least. And they were actually very consistent in the right hemisphere, such that every single participant showed these regions in the right, but you can see it's much less consistent in the left. Excuse me. So now that we found that there are these regions of sensitive spatial relations of features, we asked how selective are these regions? And we did this by examining the response of these regions to other properties we know the dorsal pathway is sensitive to. For example, we tested an allocentric relations localizer. So here participants had to judge whether the distance between the circle and line was the same or different across these two mini displays. And that was contrasted with this brightness judgment task. So you can kind of see that the circles are different brightnesses. We also compared them to a depth localizer, which contrasted 3D shape from 2D shape, as well as a action affordance localizer, which contrasted tools against manipulable non-tool objects. And what we found is that there's a greater response to the spatial relations localizer than these other properties that we know the dorsal pathway is sensitive to. And we found this both in right posterior IPS as well as right anterior IPS. And so together, this suggests that there is selectivity for an object's spatial relations within these regions over and above other properties represented by the dorsal pathway. And so having found these regions, we next want to know, do they actually contribute to object categorization? And so to do this, we presented participants with objects from 5 categories, each with 5 exemplars. And here participants were simply shown a stream of object images kind of one after the other. There was no specific task here. And the first thing we want to know is whether we could decode an object's category from these regions. That is, if we trained a classifier on the neural representation for 3 of these lamps, would it also correctly classify this held out example as being a lamp and not, say, a guitar or something? And what we found is that we could significantly decode an object's category in right posterior IPS, but not any of the other dorsal regions. So this suggests that at least right posterior IPS is capable, does have the information content to support object categorization. But we might ask, how does this decoding compare to ventral regions that are typically involved in object categorization? So we repeated these analyses with a ventral region known as LOC, and perhaps unsurprisingly found that this region was also capable of decoding the object's category. But we might ask, how is decoding being accomplished here? Are these dorsal and ventral regions using the same kinds of information to categorize these objects? So to test this, we can again use models to explore what kinds of visual information these regions might be using. So if these regions are categorizing objects by their spatial relations, then their response profile may be well captured by a computation model that also represents spatial relations, namely a skeletal model. And so to do this, we computed the dissimilarity between the objects using the skeletal model, which you can see here in this matrix. So blue cells in this model are objects that the skeletal model that have more similar skeletons, and red cells are objects that have more dissimilar skeletons. Their skeletons are more different from one another. So this is essentially our prediction space for how similar or dissimilar brain regions should see these objects if they're representing the objects by their spatial relations. And as a control, we also tested other models of vision, namely CorNet-S, which is a deep neural network that approximates the ventral pathway, as well as a Gabor-Jet model, which approximates early visual areas. And so to be clear, these matrices show how the models see the similarity between these objects. We also created dissimilarity matrices for each brain region and tested how well their pattern was described by each of these models. And so what we found is that the skeletal model was only predictive of the response pattern in right posterior IPS, not any other dorsal region, which again is consistent with our decoding results that found that this was the only region that could decode an object's category. But we actually also found that the skeletal model explained variance in left LOC, a ventral area. And here it explained variance alongside a deep neural network model. So now we have evidence of spatial relations being important for both areas. Does this mean that part relations might arise in LOC from inputs to IPS? Could it be the other way around? Or maybe this information is being computed in parallel. So to test this possibility, we conducted mediation analyses. Specifically, we tested whether the relation between the skeletal model and, let's see, can I gesture? Yeah. Whether the relation between the skeletal model and LOC was mediated by the response profile of posterior IPS. And what we found is that posterior IPS fully mediated this relation, suggesting that the variance explained in LOC by the skeletal model can be fully accounted for by posterior IPS. And moving these factors around in the model doesn't change the result. Meaning left LC does not mediate left LC doesn't mediate the relation between skeletal model and posterior IPS. So together this suggests that skeletal representations, for a spatial structure, may arise in LOC by way of IPS. But of course, fMRI has very poor temporal resolution, so it's hard to make firm conclusions about the directionality of these connections. And so in a follow-up study, we use high-density EEG, which has high temporal resolution and sufficient spatial resolution to dissociate parietal and temporal lobes. And what we found is that category information could be decoded earlier in the dorsal pathway than the ventral pathway. So here I'm showing you the time course of decoding across the two pathways. And if we resample these data, we found that decoding was consistently earlier in the dorsal pathway than the ventral pathway. So here I'm showing you the frequency of first decoding across the two pathways. Importantly, we found that the dorsal pathway also predicted the response of the ventral pathway. So this is a time generalization matrix showing you the correlation between every time point of the dorsal pathway with every time point of the ventral pathway. And so you might be able to already see that the correlations are generally stronger under the diagonal where dorsal predicts ventral. And if we rank order these correlations, we can now more clearly see that the majority of top correlations are where dorsal predicts ventral rather than the other way around. And so these findings suggest that spatial representations in the ventral pathway might arise from dorsal input, not the other way around. And so we kind of have formulated these into a more general framework, which we described in a somewhat recent text paper. This is now less recent. But essentially, we draw on evidence from different kinds of neuroimaging modalities, modeling, and patient work to argue that the ventral pathway may be better described as a basis set of local features, much like the feature space of a neural network, and that global shape properties may arise by way of the dorsal pathway. And so we suggest that a complete object representation might be achieved through the interactions of dorsal and ventral pathways. And so to kind of come back to some of the stuff that Alan is working on, there's this question of, if shape is so important, why is the ventral pathway not doing it, right? It's kind of strange, and if shape's so important, why aren't models not, using this cue? And so what we're suggesting is that we learn local features with a lot of exposure, right? So deep neural networks and the ventral pathway is very good at using these local features when the objects are familiar, when they're overlearned from the environment. But when we're first starting to learn an object category, such as early in development or even in adulthood, that's when you might need those shape representations in the dorsal pathway input. So it's kind of the interaction of the two. All right, so this shows, together these results suggest that the dorsal pathway may compute an object's spatial structure and use this information to support categorization. And I'll pause there for a second. Yeah, Mike.
So what do you think it is about the dorsal pathway that's supporting it? Sense of global shape that demonstrates that. Is it an architectural difference or even inputs?
Yeah, well, we know the inputs are definitely different. Yeah. Well, yeah. We know the inputs are definitely different, right? So there's more inputs to the parietal cortex from magnocellular pathway, right, which is generally larger receptive fields, blurrier, right? So that blurriness is nice because actually just averages out a lot of local features for you, right? So it kind of just leaves you with the spatial structure, whereas the ventral pathway gets majority of its inputs from parvocellular cells, which are much finer detail, much smaller receptive fields, which is good for that kind of color, texture, local feature information. So certainly the inputs we know already help split this information. I think there's this general question of computation though, where just because the inputs are different, that doesn't give you a similarity space of why some spatial relations are more similar to each other than the other, right? It tells you that, okay, I've already got rid of other kinds of information, but now how do we actually say where things are relative to one another, right? And I think there's some ideas about how that could work, but I think that is less clear. I don't have quite a mechanistic account for how that part happens. The architecture is just wrong. Yeah.
Time course of the
Yeah.
training Alex meant from scratch to actually start to learn global structure, and then it goes in.
Does
Right?
it?
Never
It
learns
just
it.
never learns it. Yeah, and so people have in the modeling world have really tried to like really tried to like forcibly implement these kinds of cues. So I showed you we tested models where they randomly vary the local features, right? Try to decorrelate that information, and they just learn new different kinds of local features, right? People have done this infant kind of training where they train blurry to clear, and you get larger receptive fields. You kind of do get more global properties, but then you just have a larger feature, right? It's never quite maintains a sense of where features are relative to one another. And I really think it's an architectural issue in the model. Topographic models maybe could. There's some, there might be a mechanistic reason I could go into if we like for why topographic model could learn it because it maintains that spatial map. So most models aren't random topic, right? So that's part of it. The whole brain is, yeah, that's true. But the ventral pathway doesn't seem to be learning this, right? Ventral pathway is random topic, but doesn't still seems to maintain.
Basically showing what you define as LC regionally pushed around.
Yeah.
Granularity.
Yeah.
The scrambling.
Yeah, totally. Well, and that's basically what the Jagadish paper is showing, that at a certain spatial scale, that scrambling isn't really scrambling anymore. So sorry, is it learning it locally?
Yeah, so like is it a receptive field within that receptive field it learns?
Yeah. Well, okay, so just since we're on this, I'll just jump to it. This is not part of the this is director's cut. Okay, neurally plausible. So this is, I kind of incorporate this for skeletons, but this is probably true for just thinking about spatial relations in general. So, at least in early visual cortex, we know we have, there's evidence for border ownership cells and grouping cells, right? So border ownership cells sensitive to the edges of a figure, and then they pool into these next level cells called grouping cells, right? And at least from simulations, and there's been a little bit of empirical work, is that people have found, or specifically Van der Heijden has found, that the response of these grouping cells now corresponds to the center, the medial points of a shape, which is basically like a skeleton, right? So perhaps that is what's giving us something like a skeletal representation, a spatial representation. And so this is just a computational implementation of that. So if you create a simple model with kind of edge detection neurons and then grouping cells, it also gives you a skeletal model. So this is like kind of a biologically plausible implementation of a skeletal model. And so you could imagine, and actually in some, I'm not presenting here, but in some early fMRI work that I did when I was still in grad school, we actually found that V3 coded the skeleton, right? So that was, we were expecting high-level object regions, but actually we found that this started in V3, which lines up nicely with this work. And so you can imagine a hierarchical organization where V2, V3, you have local skeletal structure at the space of parts of like, these edges are across from these edges, right? And then you move that up to posterior parietal cortex, and now you have it in a larger space where these grouping cells are now grouping up with these other grouping cells to create the whole figure, right? So that seems like a very reasonable hypothesis for how this could be implemented. I don't know, what I can't explain yet is why doesn't this happen in the ventral pathway, right? And so maybe it is just the input, and the principle is the same, just at different levels of resolution. Yeah, I don't know. Definitely.
Across time, like frame to frame, like every input's going to be different. Whereas like with the larger, like that's how you get the motion, like all of that
with magniceller.
Across multiple frames of time, you're getting more similar global structure rather than like.
I mean, that certainly lines up with our EEG data and some of the other EFIS data that shows that object information can be decoded earlier from dorsal pathway than ventral pathway.
Larger receptive field and more like video information.
I mean, that's the baby models are basically trying the larger receptive field route. Videos, I don't know that anyone's tried that. I think, again, this is basically giving you a topographic map, right, across layers, and most neural networks don't have that, right? You can almost think about transformers as almost implementing that in that they have long range connections between pieces of the image. So you can imagine they could learn those relations, that this piece over here relates to this piece over here. They don't seem to. That might be because we haven't done a fully cross design where we train a transformer with blurry input with this and this and this and this, right? But they don't seem to, which is why I'm kind of thinking right now, thinking about how a topographic model potentially could, at least has the circuitry, this kind of processing that theoretically it could given the right inputs. So, yeah.
That this being a smooth motion of an object is important. Like, isn't there research that shows that?
Mhm.
And so then to Emily's point, wouldn't that point toward the dorsal structure of bigger fields that are doing that motion integration?
Yeah, there's 100% really strong evidence that motion is crucial to forming invariant object representations. In infants, in newborn chickens, control-reared chickens, and it has to be slow, continuous motion. Seems to be really, really important. Yeah, I think that's certainly one type of input, but at some point we abstract away from that, where we no longer need that motion, right? So maybe it helps wire up the system, but it's not the cue we need to accomplish the task, if that makes sense. There's also this thing that, we're kind of bouncing around, where we've sort of moved away from the dorsal pathway as being the motion sensitive pathway, right? And that's now more of a lateral pathway. So posterior parietal cortex is probably motion sensitive, but it's not doing motion integration, right? Again, not to say they can't be communicating, getting inputs from each other, right? But it might be different scaffolded processes. Yeah. Cool. Any other questions? So this stuff has not been fleshed out in the literature by any means. So this is kind of a proposal for how this could work, potentially. Okay, so we found that the dorsal pathway seems to compute an object's spatial structure, and it can use that information to support object categorization. Um, so given that I've now shown you that infants categorize objects by their spatial relations and dorsal pathway plays this role, and also kind of offhand, I've sort of said this already, one reasonable hypothesis is that the dorsal pathway might also develop early to support these kinds of abilities. And so in this final set of studies, we asked how do the neural pathways underlying object categorization actually develop? And specifically, what is the developmental trajectory of the dorsal pathway? Might it develop even earlier than the ventral pathway? So let me tell you how we've been testing this question. So if the dorsal pathway develops earlier than the ventral pathway, then we might predict that the network organization of the dorsal pathway will already be more adult-like than the ventral pathway in newborn human infants. So here we've tested this question by using newborn fMRI data where infants were scanned while they were asleep. So these were infants that were scanned within a day of life. And specifically, what we did is we used resting state fMRI data from the DHCP, and we extracted the resting functional connectivity the resting state signal from 25 visual areas, and then simply correlated them to one another. And what we found in this pattern of correlations is that these regions already showed evidence of adult-like pathways and a hierarchical structure. Specifically, you can already see that there's these distinct groupings for early visual areas, ventral, lateral, and dorsal pathways. And this organization is overall very similar to adults. And what I think is even more interesting about this is that if we were to zoom in on this, you can also see that it's hierarchically organized in newborn infants, such that the representation goes from V1V, V2V, V3, all the way up each of the hierarchies, right? And then even splits into ventral and dorsal pathways, so on and so forth, right? You can sort of go V1D, V2D, V3D, up and down, right? And so, I love looking at this because it really just helps you appreciate just how similar the organization of the neonate visual system already is to adults. Meaning that we're born with our large-scale structure already in place. But importantly, we can now use these data to ask how adult-like are each of these individual networks. Essentially, is the connectivity pattern, the correlation pattern of dorsal regions more similar to adults than, say, other networks? And what we found is that, yes, the pattern correlations of the dorsal pathway is already more adult-like than ventral, lateral, and even occipital networks. So what this tells us is that even at the earliest stages of life, on day one, the dorsal pathway may be more mature than the ventral pathway. And we can actually go one level further with these data. Specifically, we can ask, when does this structure emerge over gestational time? And we did this by splitting the data into preterm, early term, term, and post-term infants. So these were infants that were scanned at these different gestational ages. And so if we start with the term infants, we see a pattern that's very similar to what I just showed you with this hierarchical three-pathway structure. And similarly, the post-term infants, so these are infants that are a little overcooked, also show sort of an adult-like structure. But if we go earlier in time, earlier in gestation, we start to see that this organization gets more and more disorganized, such that at early term, there's a little bit more intermixing between lateral and occipital areas. And the preterm infants show a very different kind of organization um, than the adults. What's interesting here is that you can actually see that the dorsal pathway generally it maintains its structure, right? If we zoom in, we can see that it's still hierarchically organized. And if we analyze these data quantitatively, we can see this pattern, that the extent to which these pathways group together increases as a function of gestational time, as does the maturity of these pathways. And you can see that the dorsal pathway is consistently the most mature network, and the ventral pathway here is consistently the least mature, even into post-term. And so this again tells us that the dorsal pathway may already be is already more mature than the ventral pathway. It may be able to support this kind of rapid categorization ability. Um, and so this finding leads to, I think, another really interesting prediction. Specifically, if the dorsal pathway matures so early, we might also predict that the dorsal pathway would show less functional reorganization than the ventral pathway after brain surgery in childhood. And so the idea here is that we know that the brains of children are generally more plastic than adults because they're immature. They still have capacity to change. But as I just showed you, different regions mature at different rates. So if there's also regional differences in the development of different pathways, we might predict that these pathways may be more or less able to adapt following damage or surgery. So I've tested these questions with a really rare group of patients that had hemispherectomies as children. So these are children that had, half their brains removed to treat otherwise drug-resistant epilepsy. And so kind of as an aside, I like to say how, these children are pretty remarkable that on most functional day-to-day skills, they're actually not very impaired. Many of them go to college, they ride horses. It's really a remarkable outcome for what is like a crazy surgery. But it also gives us a nice opportunity to design an experiment. So let me tell you what we did. So one property of the brain is that some functions are lateralized. For example, as I showed you in the very beginning, global form is lateralized such that it's represented more strongly in the right hemisphere of the dorsal pathway than the left. And there's other evidence of lateralization in the dorsal pathway, such that tools are represented more strongly in the left hemisphere of the dorsal pathway than the right. And what this typically looks like in the data of controls is that you get more selectivity to tools in the left hemisphere than the right, and more selectivity to basically what I showed you earlier, to global form spatial structure in the right hemisphere than the left. And conveniently, there's also lateralization in the ventral pathway, such that words are represented more strongly in the left than the right, and faces are represented more strongly in the right ventral pathway than the left. So this is kind of the typical pattern of responses that we might get in healthy children or adults. And when we've scanned new sets of controls for this study, it's basically what we get. Tools represented more strongly in the left hemisphere, left dorsal pathway of controls, alongside, tools and words, both stronger in the left side. Global form and faces more represented more strongly in the right side. And so now we might ask, what happens if we were to lose an entire hemisphere? Would the functions of the dorsal ventral pathway reorganize to the contralateral hemisphere, right? With tools and words now appear on the right side. So to test this question, we tested an equal number of left and right hemispherectomy patients. So these were patients that had surgery as young as 1 year of age, all the way up to 16. I'm sorry, all the way up to 13, and they were tested basically from 12 to 37, right? So it's a wide age range of surgeries and wide age range of when they were tested. And so before I show you the results, let's examine what the data should look like if there is evidence of reorganization. So if there's evidence of reorganization in the dorsal pathway, then for tools, we should expect patients with only a right hemisphere, here in blue, to show tool responses that are comparable to control left. Um, and similarly, patients with only a left hemisphere should now show global form responses that are comparable to control right. Right? This would what reorganization function to the opposite hemisphere would look like. And similarly, if there's reorganization in the ventral pathway, then again, we should see words arise in patients that only have a right hemisphere, and faces arise in the left hemisphere of patients that only have that hemisphere. And we might otherwise see normal responses for the categories that are already in that hemisphere, right? Patients that still have their left hemisphere should maybe show normal responses towards, okay? So no specific predictions there. So what do we find? Uh, we found no evidence of reorganization in the dorsal pathway. Tool responses for patients with only a right hemisphere did not resemble control left, and global form responses for patients with only a left hemisphere did not resemble control right. This suggests that there was no evidence of reorganization in the dorsal pathway. So what about the ventral pathway? Here we found evidence of reorganization. We found that patients with only a right hemisphere did show word responses that were comparable to control left, and patients with only left hemisphere showed face responses that were comparable to control right. So in contrast to the dorsal pathway, there was evidence of reorganization of function in the ventral pathway. And together, these findings suggest that the dorsal pathway may be less capable of organization because it matures so earlier, right? It's already close to its adult state, which makes it less malleable, less plastic when this kind of damage occurs. So again, these provide converging evidence dorsal pathway may mature earlier than the ventral pathway. Um, and so together, what we found is that using kind of classic methods from developmental psych with computational modeling is that object categorization in infancy may be supported by a spatial representation of shape, not local visual features. And then next, we found that such a representation might be supported by the dorsal visual pathway, not the ventral pathway. And finally, using fMRI with infants and patients, we found evidence that the dorsal pathway might also develop early to support these kinds of skills, right? It might develop early to support your ability to rapidly learn new object categories. And so if we go back to these examples, we can imagine how we recognize objects in this context by relying on a deeper representation of structure that, much like this shape skeleton. And zooming out from this kind of representation specifically, I had the opportunity to take sort of a bird's eye view about the development of object recognition. And what I think are the big takeaways from a review is that an infant's specific developmental niche, like the fact that they have blurry vision at birth, is an adaptation, is something that bootstraps object recognition. It's not a limitation. Um, and as a field, we should be increasingly formalizing our theories in computational terms because it helps us more directly test what it is that infants might be doing. And this is not only so we can use models to explore the biological and experiential constraints that lead to mature abilities, but it's also because development is the only process that we know leads to human level intelligence. And so by understanding developmental principles, we may also improve artificial intelligence and computational models. Um, and so kind of as I alluded to in the beginning, this, this work, this question of how humans generalize in the absence of extensive experience is not just specific to how we form object concepts or object categories. It's also relevant for understanding how humans form a lot of fundamental cognitive abilities. So these abilities like intuitive physics, spatial cognition, and so on, are abilities that all arise within the first year of life. And what's particularly interesting about these, seemingly fundamental abilities is that in adults, they all seem to, oops, I lost my animation there. They all seem to have their roots in the dorsal pathway, right? We can find these capacities in the dorsal pathway in adults, right? So this kind of offers a potential perspective of potential mechanism for how these abilities arise so early. And so now that I have my own lab, we're starting to look at this. And so to kind of highlight Alan over there, who's cut his hair, Alan's going to be looking at, starting to look at what principles might explain the diverse functions of the dorsal pathway, and starting to think about how we could create models of the dorsal pathway. So starting to talk about and think about these issues that we were saying of like, how can we reconstruct this? And then Sini is going to be doing the very hard task of scanning awake babies and asking what neural mechanisms might support cognitive abilities in infancy. asking how mature are dorsal and ventral pathways, and also thinking about to what extent subcortical structures might support these capacities early in development. And then where I hope we'll eventually get to is using computational models to ask about the experiential and biological constraints that lead to this organization, right? How does it get there? And then Olivia is our lab manager, who's a former Penn student, and so helps keep everything running, sort of contributes to all of these. With that, thank you.
Try and think of the relation between the behavior study you started
Mm-hmm.
and the later imaging study looking at the maturation.
Our infant stuff,
The
or the adult.
Yeah, so the first study that you had, the behavior where you
Mhm.
showed, we have data time maybe around 4 or 5 kids can start to
Oh yeah,
don't like
yeah,
if the stimulus is off on that, right? And there's still some
sure,
immature
right.
differences. Um,
Nope.
how do you think that that jives with though, like the neuroimaging data that suggests the dorsal string that might be doing the global shape is mature? Like, is there what seems like there might be a disconnect there? Is
Mhm.
there other systems that maybe are influencing?
So you're asking why isn't this already at adult levels if this is a dorsal process? Yeah, yeah, yeah. I think there's a couple possibilities. One, there might be just secondary task characteristics, task demands that might be hurting kids' performance, right? Simply getting them paying attention, getting them through the task. Once they start getting things wrong, they don't want to do it anymore, right? So there's certainly other factors at play here beyond simply their capacity to do this task. But I also think it's important to say that just because we found that maybe a coarse measure of functional connectivity in infants, it looks pretty adult-like, need not mean that the actual functions of the area is adult-like. We don't actually know yet how it responds to these tasks that adults can already do, right? And it might still be pretty coarse. It might be that the wiring and the structure is there and it's ready for these tasks, but there might be additional development that needs to happen with exposure, right? And again, we know that a lot of these skills, even those fundamental cognitive abilities, get better over time, right? But it might not be a qualitative change, right? It's not that it's a new skill; it's that it's a fine tuning of a skill that's already there. So that's one way to think about it. Yeah. Yeah. Yeah.
Um, relate to that question.
Mm-hmm. Yeah. Great question. Yeah. 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 Um, right. Right. Generally, no. So this is also a bit of a puzzle here. So just to unpack this, so we gave them these face and word tasks, and you can see their performance. And so I'm sorry, the gray dot here is controls, and then the color dots are different patient groups. And so you can see that their performance is pretty high, sometimes a little lower than controls, but overlapping. And we also gave them that task we gave to kids. They're also generally very good at this task. You can see most of the patients are pretty much at ceiling. Um, and just to give you a little bit more of a qualitative description, these patients can use tools. They can write. They can write just fine, right? They're not getting lost or anything like that. Um, so it's not. We have not yet been able to find a specific deficit that aligns with this lack of dorsal reorganization. One thing we have found that could explain this is that when we do a multivariate analysis of dorsal and ventral pathways in these patients, like some kind of decoding approach, we can decode this information. We can decode the presence of global form in patients' intact dorsal pathway. We can decode tool representations, right? So it might be that in patients at least, the representation is more distributed than it is in controls. So then that's maybe sufficient to get them through the task. But yeah, it's still a little bit of a puzzle. Yeah.
Find that the area dedicated to recognizing the skeleton form larger than standard is in adults. Is that, and is it in both hemispheres more than
So yeah, so we haven't done so. Infants have not been scanned with these tasks. So the infant neural data we have is from sleeping babies, right? So those data is just looking at how areas communicate with each other and are they already showing patterns of connections that are like adults or not. Yeah, no one's done that either yet. Yeah, yeah. So there's been very, very little imaging data of the dorsal pathway in kids of any age. What I can tell you is that by at least 8, people have shown that dorsal pathway can support object recognition abilities. So you can basically do these tasks where you correlate neural activity with task performance, right? And you do that a whole bunch across a bunch of areas, and you find that correlation with both dorsal and ventral pathways. But no one's done like a very careful teasing apart of representations of kids yet. And we've only done that with adults. Yeah, like kind of, yeah, right. And in language learning, it goes, it's mostly left lateralized, but it becomes more left lateralized over time, right? So yeah, you might find something like that with kids too. Yeah, no, it's an interesting question. Yeah. Hmm. Hmm. Yeah. 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 No, that's really fun. So, not that directly, but Liz Spelke, who you might be familiar with, has this whole idea that our not our object concept knowledge comes from an evolutionarily preserved representation of bodies and plants, right? So bodies, of course, have a very clear skeletal structure. Plants also kind of, they you can see ribs in them. They have this structure, and so she thinks that that it our representation for objects more generally is kind of bootstrapped out of that system, right? And that's originally the skeletal model was actually made for kind of biological form perception, right? And it's not just sort of been generalized. We just found out that it works pretty well for other things that don't have clear skeletons. So I don't think no one's actually done any work to say that, to actually test if that's true. Yeah. I mean, and so we could potentially test that in models, right? We can train models with just hands, infant views of hands, and ask whether they're more likely to learn a skeletal representation or something like that. Yeah. There has been work to really document infant visual experience. Um, and their hands are certainly in there. It's one of the most common stimuli, but the most common stimulus, well, it kind of varies as a function of age. Really early on, it's kind of just high contrast points in space, right? It's like bright lights in the ceiling and stuff. And then it becomes faces. It becomes lots and lots of faces. Um, and then it sort of builds out from there. But yeah, no, it's an interesting thought. Certainly, my hypothesis here is that, you know, it's not that we are necessarily born with a skeletal representations. It's that the structure of the dorsal pathway, this kind of boundary border ownership to grouping cell structure, is primitive, and that is sufficient to give you representation, right? It's an easy emergent property that's important from that structure. Yeah, so you don't necessarily need to build in a whole additional thing for this.